Gràcies per la música. Avui ens acompanyaran amb la música reiterades vegades, la qual cosa és sempre absolutament agradable. Però, en tot cas, permeteu-me començar. Benvinguts al Saló de Cent de l'Ajuntament de Barcelona. En primer lloc, president de l'Acadèmia, Don Dingwell, welcome. Professor Svante Pääbo, no sé si ho pronuncio bé. Director acadèmic del Hub de Barcelona, Acadèmia Europea, Jaume Bertran Petit, director del Museu de Ciències Naturals, Carles Larueza, regidors, regidores, amics i amigues. Benvinguts al Saló de Cent, que és l'origen de la democràcia barcelonina. Fa gairebé 600 anys que en aquest mateix saló l'antic Consell de Cent, que es reunia per governar la ciutat, ja va reivindicar davant el rei, en aquells moments parlàvem de reis i d'obispes, tenir uns estudis generals per adquirir la ciència. És a dir, el debat de la ciència o el paper de la ciència a la ciutat de Barcelona ha estat sempre molt important en aquesta ciutat. Som una ciutat de ciència i estem compromesos amb la ciència i amb tots i totes les persones que la fan avançar. En primer lloc, voldria agrair l'assistència de tots els presents i molt especialment també del senyor Don Dingwell, que és president de l'Acadèmia Europea. Professor Dingwell, President of the Academy European, welcome to Barcelona. Thank you for being us today. Thank you very much. I per nosaltres és un honor el dia d'avui poder vestir-nos de gala, i a més ens acompanya la música, per atorgar el lliurament del que és el 5è Premi Hipàtia, Europea de la Ciència Hipàtia, perquè aquest és un premi que busca i intenta visibilitzar l'excel·lència dins de l'àmbit de la ciència en el marc d'Europa i, com deia abans, potenciar Barcelona com a capital europea de la ciència. Amb el Premi Barcelona reivindica el poder de la ciència com a mecanisme per obrir nous camins de comprensió de la realitat i en el seu moment, ja fa 5 anys, varen posar el nom d'Hipàtia d'Alexandria en aquest premi per reconèixer la persona d'Hipàtia, però també el seu significat, que era transmetre tots els camps de saber dins el marc de la seva època, persona que en el seu moment va ser assassinada per les forces de la ignorància i també del fanatisme. Aquest premi s'adreça, doncs, a persones científiques i investigadores que hagin dut a terme gran part de la seva tasca de recerca en un país europeu i que tinguin una trajectòria d'excel·lència en la seva carrera investigadora al més alt nivell internacional. I crec que avui, sense cap mena de dubte, el nostre premiat concentra totes aquestes qualitats i probablement moltes més. Enhorabona a Svante Pääbo, biòleg, genetista i fundador del camp de l'estudi dels genomes antics. Congratulations, Svante Pääbo, on your award. Thank you for your innovative work in sequencing the genomes of extinct species and populations, and for your revolutionary contribution that have transformed the understanding of human evolutions. Today, the city of Barcelona recognizes your value and your talent, a city that has a profound connection with you. Your collaboration with the Institute of Evolutionary Biology have left a mark on the city, a city that has a profound connection with you, as well with your relationship with Carles Larueza Foix, director of our Natural Science Museum of Barcelona, is international renowned. Thank you for your advancing science and for making humanity grow, because today we need science more than ever, and we need to protect science probably more than ever. 2,000 of the world's top scientists have denounced the real danger posed by Trump administration. They are warning us that the science system is being destroyed. In the face of savage cuts and censorship, Barcelona extends its hands of all those scientists who today find themselves threatened. Estarem al costat, seguint aquestes paraules en anglès, estarem al costat del govern de la Generalitat per acollir tot el talent que avui decideixi marxar a l'altra banda de l'Atlàntic, sense perdre el nostre objectiu, i és que el primer que hem de fer és potenciar el nostre talent. Aquest és el principal talent. Però dit això, nosaltres creiem en el programa Catalunya Talent Bridge i creiem sincerament que serà un èxit. Donarem suport, com no pot ser de cap altra manera, també a l'iniciativa Choose Europe de la Comissió Europea. I ho acompanyarem amb aquells instruments que la ciutat de Barcelona en aquests moments té, sigui l'International Welcome Desk o el Barcelona Digital Talent o també a través de la Barcelona Investment Office. Aquest matí, amb alguns de vosaltres, hem coincidit ja amb el Pla Impulsat, que més enllà d'un seguit de diferents estratègies dins de l'àmbit econòmic de la ciutat, entre elles la ciència, el que intenta és donar confiança assumir les responsabilitats que aquesta ciutat té i també definir aquelles oportunitats amb les que nosaltres podem treballar. Tenim els instruments per donar suport a tot aquell que fugi d'allò que podem dir asalto de la sin razón al mundo de la razón, que deia Ginés Morata en l'Acadèmia Nacional de Ciències dels Estats Units. Barcelona sempre estarà al costat de la ciència. Al segle XX nosaltres parlàvem que aquesta ciutat tenia 4 motors i sempre parlàvem del port, l'aeroport, la fira i la zona franca. I eren curiosament motors que estaven definits des d'un punt de vista físic i geogràfic. Avui podem dir que el gran motor que té Barcelona, el 5è, és la ciència. La ciència, el coneixement i la tecnologia. Per això nosaltres quan parlem de ciència parlem de cultura. Anem a Guadalajara i a Guadalajara no parlarem tan sols de llibres, parlarem també de la Bienal de Ciència a aquesta fira internacional. I per això construïm la ciutat al voltant de la ciència, sense oblidar altres sectors que creiem que són importants. Per això volia acabar amb unes paraules del Richard Feynman, que va reflexionar sobre el valor de la ciència en un dels seus textos més significatius a l'any 1955. Feynman ens va, que jo alguna vegada el cito perquè és l'únic autor que quan parlava de la quàntica, deien, si tu creus que coneixes la quàntica és que estàs equivocat, no coneixes la quàntica. Però la cita aquí és molt interessant i més en aquests dies que estem en aquests moments. Feynman ens va mostrar que la ciència porta llibertat a través del dubte i l'incertesa, quan hi ha alguns que creuen que tenen tota la certesa. Ens va advertir sobre la responsabilitat que comporta el poder del coneixement científic. I ens va fer veure la bellesa intrínseca de comprendre com funciona el món més enllà de les aplicacions pràctiques. És a dir, parlant de ciència, en Feynman ens parlava de coneixement, però també ens parlava de responsabilitat i parlava de bellesa, entrellaçant els 3 conceptes. I Feynman acabava el seu text d'aquesta forma, en anglès. It is our responsibility as scientists, knowing the major progress and great value of a satisfactory philosophy of ignorance. The major progress that is the fruit of freedom of thought to proclaim the value of this freedom. To teach how doubt is not to be feared, but welcomed and discussed. And to demand this freedom is our duty to all coming generation. Aquest serà el nostre desafiament, no tenir por del dubte com un motor per poder transformar la nostra societat. Creure que l'ignorància, d'alguna forma, pot ajudar a vegades a través del dubte a intentar ser una societat millor. En tot cas, visca la ciència i visca Barcelona. Moltes gràcies.
Gràcies. Benvingudes i benvinguts. Welcome to the award ceremony of the Hipàtia European Science Prize, an award that recognizes the best career of a person in the scientific field in Europe and puts Barcelona at the center of knowledge and research. Today, at this event, we will grant the 5th edition of this award. But first, a few words from the president of the Academia Europaea, Professor Don Dingwell.
It's a great pleasure to be here on behalf of the Academia Europaea, which, as you see, is a part of everything that we're undertaking today. And it's a very proud honor that the Academia Europaea carries to participate in the administration of the quality of the selection of candidates for the Hipàtia Prize. My name is Don Dingwell, and I've been acting president from the 1st of January of the Academia Europaea, and it's a special honor for me to be able to be here today. Barcelona is one of the most natural homes of the kind of science that the Academia Europaea is trying to propagate throughout Europe and trying to maintain in its stature in our society. And whenever we come to this wonderful city, we realize how much at home we are immediately as scientists. Not as tourists, not as football fans, but as scientists. I'm living in Munich. I see lots of parallels between Munich and Barcelona, and so I think I can speak a little bit with authority on all 3 of those fronts. Congratulations from my side, of course, to our prize winner, Svante Pääbo, who is, of course, doing us a great honor by his presence today and by the contribution that he will make to us. It has certainly been an exciting result of the process that we have employed to find the Hipàtia Prize winner for this round of selection, and of course much more will be heard about and from Svante in a few minutes. I'd just like to use the 2 or 3 minutes that I have to touch on 3 words, recognition, freedom, and reputation. Recognition, freedom, reputation. First of all, for recognition, that's what we're doing here today. We do not have annual financial reports to impress Wall Street with how much good we are doing for the world. What we have is recognition of the scientific achievements that go far deeper than the daily, weekly, monthly economic impact of our activities, and which lead us into the years and into the decades to come. And it's this world that we all live in as scientists, that we look at the mid to long-term impact of what we do. And of course, if we're asked to quantify and measure the impact of what we do, we all fail miserably, serially, because it's essentially an impossible intellectual task. But one can grasp with some experience what kind of impact science has, and there's no question that in the person and in the work of Svante Pääbo that we have somebody who's had a massive impact already, and we will see it unfolding in the years to come. The second word I mentioned was freedom, freedom of research, and I'm always proud as somebody who has obtained the German citizenship to remind you that in the Constitution of Germany, the basic law, the freedom of research is ironed into the basic law. So when they wrote this down after the Second World War, they took the time to come up with the phrase, the freedom and the concept, the freedom to do research. What does it mean? It means that if you are doing research, you must do it in an open system, free from external constraints, censorship, or too much mission-driven or directional advice, which is coming from people who are not practicing scientists. And in this ecosystem and in this environment of being able to work freely, we see some of the greatest value of what we can do as intellectuals working in research. And we hope that the European Union, of which Barcelona is such a very important element, will be in some way listening to us today in its development of plans for the next framework program, which will ensure that a significant fraction of funding for research is applied to the freedom-driven research, if you will, rather than simply the mission-driven research, which is certainly on everybody's tongue these days. And my last word was reputation, and I turn again to the location that we are in today. It is the reputation of Barcelona as a city of science that we want to protect and that we share the enthusiasm for with you. And I think that together we can withstand the competition that we have from some of the other famous aspects of this city simply by maintaining the effort that we have to keep Barcelona at the front of international sciences. I congratulate you. Thank you very much.
Ara el que farem és parlar una mica sobre la música que avui escoltem aquí, perquè no és una música a l'atzar, neix de la sintonia entre el Carles, el músic que ens acompanya, i el premiat de l'Hipàtia 2025. As you will have noticed at the beginning of the event, we will be accompanied by 3 musical performances by Carles Marigó. Carles is a pianist, composer, and professor of improvisation at the SMUC. Trained in classical and contemporary music, he has boldly explored the limits of the piano, integrating improvisation, electronics, and sound experimentation. All this made him one of the most original emerging figures in the Catalan music scene. The first piece was an improvisation on an 18th century song, very popular in Sweden. This melody, so present in the collective memory, seems almost inscribed in the country's sonic genetics. Now, we will listen to the central music piece, an original composition written especially for the occasion. This work originates from the dialogue between the musician and the scientist, Dr. Pääbo, who shared a video shot in Okinawa while he was immersed underwater. From this personal material, Carles Marigó has created a piece inspired by breathing, both in the literal and symbolic sense. breathing as a vital act, as an invisible movement that sustains life. The piece, Piano Portrait for Svante Pääbo, does not have a melody in the traditional sense. Nomen Zim is presented nor developed, but rather an open structure based on textures and subtle variations. In a way, this sonic portrait seeks to do the same as Dr. Pääbo's research, to descend into what is deep and essential. What he explored through DNA is sought in this piece through sound. Doncs gràcies, Carles, per aquesta peça. Thanks for the music. Now we will listen Professor Jaume Bertran Petit, Academic Director of the Barcelona Knowledge Hub, the hub of the Academia Europaea, and President of the jury that awarded this edition's prize. You can go outside.
Molt bona tarda. Jordi Banys, tinent d'alcalde, Joan Gómez Pallarès, director general de recerca, benvolguts tots. En pocs minuts us parlaré de 5 coses diferents. Ho faré ràpid, però concís. First, what is the Barcelona Knowledge Hub of the Academia Europea? So we know that the prestige of the Academia Europea is without doubt. I used to say that is maybe the best lobby existing for research in Europe, and the academia should also go down to countries, to regions, to cities, to the population, and have a say in research politics, as it is this case. Our mission on the Barcelona Hub of the Academia Europea is to promote scientific dissemination, built on excellence in scholarship, aimed not only to members of the academia within geographical area, but large academic community and the general public, with a special emphasis on cross-sectional perspective, where the academy may have more strength, because it goes from natural exact science, social sciences, and humanities. Envision is in turn science into the shared common value among all citizens and to become a hub of thought and dialogue on global challenges facing humanities. So for all that, we are having several what we call the dialogues, one that we are doing monthly in Barcelona. The first kind of dialogues is called Agora, which are scientific discussions that promote interdisciplinary dialogue between 2 experts on scientific advance that are of high social relevance. These in general are being held in Catalan or in Spanish for the general public. But we have also the other ones called Frontiers, in which we introduce new series of high-level scientific discussions in English that explore the limits of knowledge and cross-disciplinary boundaries. This series focuses on perspective of very distant fields of knowledge, for example, social or ethical or philosophical implication of scientific and technical advance. And once a year, we have a high-level, much high-level dialogue that is called Disputatio, and since it began in 2013, the hub, in collaboration with Barcelona City Council, organized this very special Disputatio that is a debate mirroring the medieval Disputatio, in which we focus on very, very interesting points, as in the last few years, the origin of human violence or the rise of misinformation. We have also some activity on arts and sciences aimed at exploring and developing synergies between these areas. So we try to enrich the dialogue that has begun between artists and scientists, and we are having some impact. For example, the inclusion of these sessions of art and science in the last building bridges, and will be also quite important in the next one that this year is going to be held in Barcelona in October, in which we will have the participations, among others, in a scientific meeting of Jordi Savall. And last, but very important, we organize with the Barcelona City Council the Barcelona Hipàtia European Research Prize, and we are here to celebrate that as the recognition of outstanding researchers conducted in Europe. So these are the activities we are doing. And these activities, I have to say also, are being done thanks to the strong help of Maite Sanchez, that is here, that is the hub manager of our institution. And of course, the second point has to be the acknowledgements, agraïments. Agraïments a l'Ajuntament de Barcelona, especial a la tinència d'alcaldia de Jordi Banys, amb l'Anna Brony al davant com a interlocutora, a la Conselleria de Recerca i Universitats de la Generalitat de Catalunya amb el suport de la Direcció General de Recerca de Joan Gómez Pallarès, a la Fundació Catalana per la Recerca i el seu director, Miquel Gómez, per acollir-nos institucionalment i donar-nos la fortalesa que estem assumint. També a la Real Acadèmia de Ciències i Arts de Barcelona, la RACAP, i el seu director, Joan Isern, per acollir moltes de les nostres activitats en la seva fantàstica seu, i els membres del nostre Steering Committee del Hub, Alexander Fidora, Ramon Gomis, Genoveva Martí, Andreu Mascolell i Marcel Schwart, em penso que tots ells aquí. Gràcies pel vostre suport i la vostra guia. El tercer punt és la música, que ja ens l'han explicada, però m'agradaria que sigueu tots molt conscients que hem estat a l'estrena mundial d'una obra del nostre estimat compositor Carles Marigó i que es titula així Piano Portrait of Svante Pääbo, i m'agradaria simplement assenyalar que vaig ser testimoni del diàleg entre ells que varen portar-los a buscar músiques i sonoritats preferides. Per un costat, músiques tradicionals o la sonoritat pròpia, íntima, personal de la immersió de Svante sota el mar. A partir d'aquí, Carles Marigó ha fet servir les seves dots innovadores i d'improvisador, de fusió de tècniques clàssiques i contemporànies per construir variacions sobre els tres temes que escoltarem. This musical tribute enriches our celebration, creating a profound dialogue between art and science that reflects the spirit and vision of the Academia Europea. The fourth point is on the sculpture of the prize. The Hipàtia Prize given today represented by a sculpture of Sergi Aguilar that is here with us. Aguilar is one of the most prominent contemporary sculptors in our country, with a body of work that constantly engages with space, memory, and geometry. His sculpture for the Hipàtia Prize is not only an object of great formal beauty, it also symbolizes, it's a symbol of inquiry, balance, and creative tension, values that resonate with the spirit of the Hipàtia and with the mission of the academic community. And last, to finish, on Svante Pääbo having had the prize. His revolutionary research has profoundly transformed our understanding of human origins, illuminating fundamental aspects of what truly makes us humans. Widely recognized for his seminal work on extinct hominids, I would like here, before the laudatio that is going to be done after, to emphasize two key contributions that I personally think of special influence. I'm sure you are not guessing which ones are going to be. The first, the concept of the mosaic structure of the genome. Long time ago, you may not remember, you wrote a paper on the mosaic structure of our genome, where he introduced the view of the genome as a mosaic composed of discrete segments, each with its own distinct history and connections to diverse contemporary ancestral populations. This groundbreaking perspective has enabled deep analysis even at the level of individual genomes. Thanks to this vision, now we can reveal intricate footprints of human history encoded sometimes within a single person's genetic makeup. And the second, his conclusive demonstration of introgression in the human lineages. His work has shown that modern humans carry genomic segments inherited from extinct hominin population. But the interest is that it has opened an entirely new research avenue, allowing scientists to trace and retrieve genetic contributions from ancient, extinct groups within every genome today, fundamentally reshaping our understanding not only of human evolutionary history, but of many other species. In a certain way, be it through the retrieving of ancient DNA, be it by calculating how were the ancient genomes, now we know the history much more in depth. I have here the scores, and remember that if you want to know more on the work of Svante, he published this book, originally in English, has been translated. There are 2 editions in Spanish, and is highly recommendable. Thank you very much.
Thanks for the words, Jaume Bertran Petit. We will now proceed to present this year's Hipàtia's prize. And to do it, to introduce this year's winner, we will hear from Professor Carles Lalueza, director del Museu de Ciències de Barcelona, who will deliver the award-winning lecture. Yeah, please.
Tinent Jordi Valls, autoritats, membres de l'acadèmia, Svante, amics, continuaré en anglès. It is a great honor and a pleasure to introduce the winner of the Hipàtia Award this year, Professor Svante Pääbo, who has had the generosity of collaborating with me in a number of scientific adventures over the last 20 years. I first met Svante in Oxford in 1997. He has just published what is arguably a landmark study in human evolution, the first retrieval of DNA sequences from a Neanderthal, in fact, from the very Feldhofer specimen from the Neander Valley that gave name to this group, to this human group. Neanderthals were a group of hominins that lived mainly in Western Eurasia between 500,000 and about 40,000 years ago. In the previous decade to this remarkable achievement, Svante conducted a painstaking experimental research work aiming to understand the features and possibilities of ancient DNA and trying to establish quality control tests for the scientific field. Until then, the ancient DNA field was immersed in improvisation. In this crucial sense, we can say Svante forged this scientific field himself and established the basis of what is today an extraordinary way to look into the past to learn about the future. We met again in Leipzig in March 2005 as a consequence of the first genetic results from an Iberian Neanderthal coming from the cave at El Cidrón in Asturias. He was already established as director of the Max Planck Institute at Leipzig, where he conducted the most significant part of his career. 1 year later, Svante started the Neanderthal Genome Project, an ambitious scientific project with the goal of obtaining the whole genome of one of these extinct hominins. To put this project into the historical context, it is worth remembering that the Human Genome Project, that took more than 20 years, had just been finished 5 years ago. Also, when he started this project, the sequencing technologies needed to do it were not even invented. Besides an extraordinary scientist, you have to be a visionary to launch a project like this. It didn't look as something possible, and yet, using new sequencing technologies that emerged in 2008, he was able to culminate it in 2010. This was not only a scientific revolution in the field of human evolution, it was also the beginning of paleogenomics, an absolutely new field to study human origins. One of the most surprising findings was the discovery that although Neanderthals were extinct, all modern humans outside Africa carry in our genomes a small but significant portion of Neanderthal genes. This is the result of an intermixing event that took place between 45,000 and 49,000 somewhere in the Middle East. Incredibly as it may seem, before the end of the same year, 2010, Svante was able to produce a second genome from yet another hominin lineage, the mysterious Denisovans, named after a pinky bone found in a Russian cave of Denisova. These Denisovans also contributed a substantial fraction, genetic fraction, to modern Oceanian individuals, leaving again an indelible mark in their genomes. Some of these Neanderthal and Denisovan genomes that help our ancestors to quickly adapt to the Eurasian environments also influence the physiology of modern humans. In some cases, these genes affect our immunological response to some infections, including the COVID-19. The Nobel Committee understood that looking to the evolutionary background of genes associated to health conditions can help understand their treatment also. For this research, Svante has received many awards, to name just two: the Nobel Prize of Medicine and Physiology 2022, or the Princesa de Asturias de Investigación Científica y Técnica, granted 5 years before the Nobel in 2017. These studies represented a change in paradigm in a human evolutionary process that no longer can be conceived as a simply branching process. We have to imagine evolution as a complex network, interconnected at multiple levels and in multiple places. The landmark of the year 2010, which can be considered for the ancient DNA like the Annus Mirabilis, was a technical tour de force, but above all, it was a conceptual turning point. To put it simply, the impossible was made possible, and in fact now is even routinary, and quite a few laboratories across the world generate thousands of genomes from the past at an exponential rate. The handmade techniques Svante conceived and started are now industrialized, and what was excruciatingly costly laboratory work has become now a commodity, with perhaps some bulky computational work. I suspect Svante will agree with me that the notion of adventure into the unknown is somehow lost. But this is how science progresses. And progress, in this case, relies in standing upon the shoulders of a giant, and not because he's just a tall person. Svante has developed almost all his career in Germany at the Max Planck Institute. It is worth remembering this institution has recognized his talent and has supported his research. This is also a lesson for us. Talent is scarce by definition, and it needs to be supported. And it's not only a question of money or resources, que també. If we want one day to have someone able to conduct here in our country disruptive science such as this made by Svante, we need to attract talent, support talent, make things easy for talent. And finally, I would like to conclude stating that it has been a deep honor and a source of happiness for me to collaborate with Svante along these years, and it's also an honor that my home city, Barcelona, has recognized him with the Hipàtia Award. Thank you.
Thanks for the words, Carlos. We will now proceed to read the verdict of the jury, which met in Barcelona in the 17th of October 2024. Is this. The members of the international jury unanimously agreed to propose as winner of the 5th edition of the Barcelona Hipàtia European Science Prize in the field of life and health science, Professor Svante Pääbo, for his groundbreaking work in sequencing ancient genomes, which has revolutionized our understanding of human evolution and provided invaluable insights into the genetics links between extinct hominids and modern humans. Now, the Deputy Mayor and the President of the Academia Europaea would come to the stage, please, and present the award to the winner. And please, Professor Pääbo. Congratulations, Professor Pääbo, and the winner is now to read some words for us.
Well, Deputy Mayor, President of the Academia Europaea, Rector Magnifica, dear colleagues, dear friends, thank you very, very much for this honor bestowed upon me today. And thank you especially also for me moving and very memorable music and art that has been created for this occasion. It's also especially a happy occasion for me to receive this honor in the city, in Barcelona, that over the past decades have developed so remarkably into a center and a hub for biosciences in Europe. So thank you very, very much. Thank you. So what I then wanted to do over the next 45 minutes is to tell you then a little bit about our efforts to sequence the Neanderthal genome, talk a bit about what we are in the process of learning about the history of Neanderthals and Denisovans, and then in the end talk a little bit about how these now extinct forms of humans may influence us today. But before we then do that, I want to just remind you about something you all know, and that is that if you study genetic variation, if you sequence genomes of people from all over the world, is it a problem? I should stay by this, maybe. If you sequence genomes from people from all over the world, you will find something surprising, and that is that most genetic variation exists in Africa. And if you take everybody outside Africa, although many, many more people, of course, live outside Africa than inside Africa, there is less genetic variation there. Because there is sort of a component of the genetic variation that exists in Africa that is not seen outside. An interpretation of that is that the ancestors of all present-day humans evolved in Africa, lived there for a while and accumulated genetic variation, and then a part of that variation, if you like, went out and colonized the rest of the world. And then with genetic tricks, we can figure out approximately when that exit out of Africa was, and it was quite recent in evolutionary terms, for sure less than 100,000 years ago, more like 50, 60,000 years ago. And an interesting thing is that this is very recent. So there were other forms of humans around at that time. There were other forms of humans in Africa, and quite famously outside Africa also, most famously then in Western Eurasia, Neanderthals that existed there since 500,000 years ago or so, and other forms in Eastern Eurasia that we are beginning to learn more about only. So why should we be interested in Neanderthals that are not here anymore? There are several reasons, I would say, for that, but two of them are that they are the closest evolutionary relatives of everybody on the planet today. So if we want to define ourselves from a genetic, biological perspective as a group, it's them we should compare ourselves to. How are we similar to them? How are we different from them? And another reason is that they were here quite recently, until, as we heard, 45,000 years ago or so, maybe 1,200, 1,400 generations ago. So an interesting question is what happened when our ancestors met them? How did we deal with each other? But to now do this, ask these questions from a genetic perspective, what we then need to do is retrieve DNA from bones that are at least 45,000 years old and sometimes much older than that. And that's then what our laboratory is sort of struggling with since 35 years or more. And this all started in the early 80s then, when we sort of realized that there are all these thousands of mummies in our museums from ancient Egypt that look very well preserved, like this mummy, which is 2,300 years old. So it was sort of a rather obvious thought to see, could we extract DNA from tissues of such remains and retrieve genetic material, retrieve DNA? So a reason to also be hopeful with that is that you can say in the skin of such an individual from that mummy there, see structures that are cell nuclei where the DNA would be preserved. So I went ahead and did that with great enthusiasm in the lab next to my other experiments, retrieved some human DNA, published it to great fanfare in Nature, and then over the next 5, 6, 7 years, it progressively became very clear that the DNA sequences I had retrieved there for sure didn't come from the mummy. Perhaps they came from some archaeologist, from some museum keeper, or from myself, because what I didn't realize was that there were a lot of technical problems with this. And those problems come from the fact that such old DNA, there's very little of it there, often 100,000-fold, 1,000,000-fold less than in fresh tissue. The DNA is degraded to very short fragments, it's chemically modified, and is present in a large excess of bacterial DNA and fungal DNA that are there in much, much larger amounts than the endogenous DNA. So one consequence of this is that tiny little amounts of contamination that can come into your experiments from a dust particle in the room, for example, would not at all influence your work if you study present-day DNA, but when you study these ancient extracts, it may totally overwhelm your experiments, and that's sort of what had happened to me. So over the next couple of years then, there sort of was a mania of cleanliness that took over our group, where we tried to avoid contamination by working under special conditions and special hoods, dressing up in funny clothing, etc. We also shifted to working not with human remains, but with extinct animals, because it's a lot easier if you study, say, remnants of a mammoth. If you find something elephant-like, you're quite convinced that you have the right thing. Whereas if you study an ancient Egyptian mummy and it looks human-like, it can be difficult to know if you have an endogenous DNA sequence. So we worked on sort of all steps of you extract DNA under clean conditions, so you make it into a form that you can sequence efficiently. And as we heard, the human genome came along, so we were able to start comparing, say, if we studied human remains to where small fragments might come from in the human genome. So we got a lot more efficient in this. The sequencing technology got a lot more efficient. So we could sort of start dreaming about studying the genome of the Neanderthal. And one part of that was then to try to find archaeological sites that were particularly well preserved in terms of how DNA is present in the bones. We settled on one site in Croatia, Vindija Cave, used 3 different bones from 3 different Neanderthal individuals from that site, sequenced over 1 billion DNA fragments, and then with computer approaches developed largely by Janet Kelsey in our group, sort of matched these short fragments to the human genome, taking into account that there are errors, typical errors in there induced by chemical modifications that accumulate over thousands of years. So then, as we heard in 2010, we had a first draft version of the genome, a very rough version. We had covered a little over half of the part of the genome we can map these short fragments to, and we could begin to ask questions. And one of the obvious questions we were interested in was what happened when modern humans came out of Africa, met Neanderthals, did one mix with each other or not? And if one did mix with each other, we would of course expect that Europeans today would share more genetic variants with Neanderthals than people in Africa, where there had never been Neanderthals. You would not expect Africans to have any special relationship with Neanderthals. So we put together a consortium of population geneticists from different places asking these questions. We had the Neanderthal genome there. We went out and sequenced from present-day people 5 individuals from different parts of the world. At the time, taking care of sequencing it ourselves to have exactly the same types of errors and things like that in these different genomes. And asked a very simple question. So if we just take 2 present-day individuals, and to illustrate it here, 2 Africans, and find all the places in the genomes where they differ from each other. And then we take the Neanderthal and say, how often does the Neanderthal match one African or the other African? And it should be 50-50, right? Because Neanderthals have never been in Africa. There's no reason to think that one African would be closer to the Neanderthal than another African. And indeed, statistically speaking, it's 50-50 matching. Then we did a sort of crucial experiment, taking a person from Europe and from Africa, asking the same question. And to my big surprise at the time, we actually found significantly more matching to the European individual, suggesting that there might have been a genetic contribution from Neanderthals to the ancestors of present-day Europeans. Even more surprising was that we took a person from China and from Africa. We again see more matching there. Although most people would say there had never been Neanderthals in China. If we do the same thing in Papua New Guinea, where everyone would agree there had never been Neanderthals, we again see more matching. So in fact, wherever we look outside sub-Saharan Africa, we find this extra matching to the Neanderthal. And the interpretation of that sort of as a big overview, it still holds up, is that we imagine that when modern humans come out of Africa, they meet Neanderthals early on. We know there were Neanderthals in the Middle East, and if these early out of Africa modern humans mixed with Neanderthals, they can sort of carry with them this contribution from Neanderthals out to the rest of the world, also parts where there have never been any Neanderthals. And the result of that then is that if your genetic roots are outside Africa, something like 1 or 2% of your genome comes from Neanderthals. So what has happened in 2010 is that we have gotten much, much better Neanderthal genomes, where we don't just have half of the things we can map these short fragments to. We have mapped all those parts many times over in at least 3 publicly available now Neanderthal genomes, so we can ask more detailed questions. So we can, for example, look in a number of Europeans. Here an example of chromosome 9, and mark on the chromosome large segments of tens of thousands, hundreds of thousands of nucleotides that are identical or almost identical to the Neanderthal genome. And you will see that that then adds up to 1 or 2% of the genome. But you will also see that different individuals carry different parts of the Neanderthal genome often. So you don't carry the same parts as I do, So we can also jump from individual to individual in this room, for example, and see how much can we puzzle together of the Neanderthal genome from people here. And that's sort of suggested in blue down here. And it turns out that something like 50–60% of the Neanderthal genome is still running around on two legs today, so to say. So Neanderthals are not totally extinct. They sort of live on in many of us today. And as we also heard back then, there was another sort of discovery we stumbled over, and that was from that site in southern Siberia, a tiny little bone that is a fragment of the last digit of a pinky of a child that turned out to be very well preserved from a molecular perspective, so we could produce a good quality genome. And we were very surprised to find that it was not a modern human, it was not a Neanderthal as we had thought, it was sort of something else that goes back to a common ancestor here, shared with Neanderthals quite far back. These datings are always sort of approximate, but sort of if the divergence between Neanderthals and modern humans is something like 600,000 years ago, this is in the order of 400,000 years ago or so. So we realized we stumbled over some new form of extinct human that one didn't know about. We didn't know how they look. We don't know what stone tools they did or so on. We called them Denisovans after this site in southern Siberia in the Altai Mountains. And we could ask questions such as, have these Denisovans also contributed to present-day people? And it turns out they have. So there's no contribution in Western Eurasia here, but wherever we look in mainland Asia, there is a small contribution, less than 1%, on top of the contribution from Neanderthals that everyone outside Africa has. And in some parts in Oceania, Papua New Guinea, Aboriginal Australians, it's up to 5–6% contribution. So we are still sort of learning a lot more about Denisovans, where they existed and how their population history has to indicate that. If we look at this tree again, they are sort of the sister group of Neanderthals there when we look at the nuclear genome. And the nuclear genome, of course, is a sort of overall picture of our genetic history. We inherit DNA from our mothers and fathers, and it's an average of the genetic history back through time. But if you look on another part of our genome, a tiny part, the mitochondrial genome, that we inherit from our mothers. So it represents the female side of history and a snapshot of history only, so to say. But if we look at that from these Denisovans, we find that they don't come here, the mitochondrial genome doesn't come with the Neanderthals, but much further back in time, about 1 million years or so ago, which was very surprising. How did that fit together? And the perspective of that we're actually getting from work that we do together with people in Spain, Jean-Luis Arzuaga and his team at Atapuerca, where they find these amazing fossils that are very old, perhaps over 400,000 years old, from this amazing site, where we have been able to retrieve small amounts of nuclear DNA. And if we're sort of asking to what group that nuclear DNA comes from, it clearly comes mostly from Neanderthals. So sort of this Sima hominins sort of are early on the Neanderthal lineage. But if we now look at the mitochondrial DNA from Sima de los Luesos, we again find something very surprising. The mitochondrial DNA there comes with the Denisovans falling outside and not together with the Neanderthals. So it seems that the mitochondrial DNA will sort of come over to Neanderthals from something related to early modern humans. So there's, as you may know, another part of our genome, the Y chromosome, that's inherited on the male side. That is sort of the male view of history, because the Y chromosome is inherited from fathers to sons through history. So if we now look at the Y chromosome in the Denisovans, it again turns out that they don't come with the Neanderthals. They fall much further back, 700,000 years ago or so, on its own lineage. So the picture we're sort of beginning to get is that there were modern humans, there were the Denisovans, who were a sister group of Neanderthals, but there were contact here, and there were contact between Africa and Neanderthals that did not affect the Denisovans that were much further in the east. And we see that if we look at the Y chromosome as this contribution of the Y chromosome to the Denisovans to Neanderthals from Africa, and the mitochondria that is also contributed to Neanderthals here. And they have sort of then permeated the Neanderthal population. We don't know yet quite how big this contribution is in the nuclear genome. That's sort of really ongoing research where I think we will learn much more in the next 2 years. But if I just should summarize then what we think we know about the origin of Neanderthals, Denisovans, modern humans from studying genomes, Neanderthals and Denisovans have some common origin in Africa more than 500,000 years ago. Their ancestors come out of Africa and evolve in Western Eurasia to what we call Neanderthals, in Eastern Eurasia to what we call Denisovans. Where the border between these groups were, we don't know. It shifted over time, I think, but in southern Siberia, for example, we know there were Denisovans and sometimes Neanderthals at other times in the same Denisova cave. Then there is this gene flow from Africa into Neanderthals that don't affect the Denisovans, that contribute to the Y chromosome and mitochondria and some nuclear variants to Neanderthals. And then there's an origin of modern humans in Africa. They come out, mix early on with Neanderthals, something like 45 or 50,000 years ago, continue to spread out, mix several times with Neanderthals, mix with the Denisovans in the east, several times with different populations of the Denisovans, and continue to spread out to other parts of the world where these other forms of humans never came, Australia, Americas, and so on. And then these other forms of humans disappear, but they live on a bit as this contribution to present-day people from Neanderthals and from Denisovans. You may now ask, are Africans sort of fundamentally different in that they don't have this contribution from other earlier forms of humans? I don't think so. We don't have genomes yet from extinct forms of humans in Africa. But since modern humans mixed with other forms outside Africa, I would not be surprised if one did so and one spread across Africa too. So I think in the future we will learn that that also went on in Africa. And there are some indications from present-day genomes, modern human genomes in Africa, that that may have gone on. We're also getting a very sort of direct view of this mixing these days, when we go back to very early modern humans in Europe. The first case of that was a site in Romania, was a cave, where cavers a couple of years ago found this mandible that looks like a modern human mandible. It's 40,000 years old, so it's a very early modern human in Africa. So we were very interested, particularly Mattea Heidenjack, who worked a lot on these early modern humans in our institute, if this person had already mixed with Neanderthals or not. So if one looks at all the different chromosomes from this 40,000 years old individual and mark here in blue the segments that are similar to the Neanderthal genome, you will find that there are not only 1 or 2% here, there are large, large sections that are almost identical to the Neanderthal genome. And this, of course, means that this individual had a close relative in its family history that was a Neanderthal. Because the first child, if a Neanderthal and a modern human have a kid. The first child, offspring, will of course have one chromosome from a Neanderthal and one from a modern human. If you then continue to have children in the modern human groups, these Neanderthal segments will become smaller and smaller due to recombination between the chromosomes with time. So if you have large segments, it means you have a rather close relative who was a Neanderthal. And in this case, you can show that it was 6 generations ago, 5 generations ago, or 4 generations ago that this individual had a Neanderthal relative. We almost caught someone in the act, so to say, of mixing with the Neanderthals. So I thought that was a total coincidence that we stumbled over this. But then in the meantime, there's this other site from Bulgaria, Bachukiro Cave, where one finds this early technology typical of the very earliest modern humans that come to Europe. Down at 45,000 years ago, there are remains of 3 individuals from which we could retrieve DNA. Higher up, 10,000 years later, there is from another individual. So if we look at these 45,000-year-old individuals, they all have some of these very large segments, suggesting that they have recent relatives that are Neanderthals. 10,000 years later, 35,000 years ago, people look just like people today. No special relationship to Neanderthals. So in the meantime, Johannes Krause's group in the Institute have retrieved DNA from other early modern humans from 2 sites that don't have this close relationship to Neanderthals. It almost begins to seem like there's a northern group of early modern humans that didn't mix with Neanderthals, and there's a southern group that did mix with them. But the picture that is still sort of surprising to me is that it seems to be that at least some groups of early modern humans mixed quite extensively with resident Neanderthals. So if we think about why did Neanderthals disappear when modern humans came, it may not be that we need to think about grand genocides or so. It may simply be that one mixed with each other, and if modern humans were more numerous than Neanderthals, they may have sort of just been absorbed into modern human populations. But the question is sort of why is it Neanderthals disappear and modern humans remain? There are at least 4 things we could hypothesize that are not mutually exclusive with regard to this. One is this thing I already hinted at, that modern human populations were probably much larger. So if you look on the amount of genetic variations in Africa today or non-Africans, as we've said, there's less variation outside Africa. But if you look in Neanderthals, there's even less genetic variation, suggesting that there was sort of even smaller groups. And actually one of the first indications of that came from El Cidrón in Asturias, where Carlos Laluesa Fox and others together with us looked at genetic variation and found that it's sort of about 1/3 of the genetic variation present-day Europeans in these Neanderthals. There is also archaeological indication of having more archaeological sites of early modern humans than Neanderthals, and that those sites seem to have been more intensely used. There's more animal bones from slaughtered animals there, etc. So, and humans were also spread, as we've said, across open water to all parts of the world, again suggesting that they are much more numerous than Neanderthals. With time, there is also much more rapid cultural evolution that takes sort of, you can find that in that technology becomes regionalized for modern humans, where it's very homogeneous in Neanderthals over their whole range. And there are things such as projectile weapons that seem to come with modern humans. You start to see the earliest evidence of bow and arrow seems to come about the time when modern humans appear and become sort of very numerous and with that. And finally, there is figurative art. And I say figurative art because one has here in Spain cave art that goes back so far in time, so it should be Neanderthals that did it. But this Neanderthal art, when I joke about it, so to say, it looks like very modern art, because I can't see what it depicts. Whereas art that really depicts something that I immediately recognize what it is, comes with modern humans. So, to me, it's quite clear that there's something very special about modern humans, different from these earlier forms of humans. And that has made us sort of very interested in looking at genetic changes here on these lineages that happen early on, that go to the Neanderthals, but particularly things that are there in present-day humans and not in Neanderthals. Particularly things that we all share with each other today on the planet and that Neanderthals didn't have. So genetic changes that spread to everybody, or almost everybody. Because I think maybe there are functions hiding there that are really unique and maybe part of the explanation to why modern humans have had the history we have had. So look at things there that you find everywhere, make a list of that. And it's not so many things, actually. It's something like 30,000 changes or so. So if you compare two genomes that you say received from your mother and from your father, it's in order of 3 million differences. But if we make the requirement that we should all share a genetic variant today that Neanderthals didn't have, it's just something like 30,000 of them. And those that change, say, amino acids in proteins is something like 100 of them or so. So we've been very interested over the past 5, 6, 7 years in those things and have the bias, of course, to think that changes in genes that have to do with brain development or brain function might be particularly important. Because we, after all, think that there's something socially or cognitively special with modern humans compared with these other groups. So we've studied something like a dozen of these proteins with such changes. I just want to give you a flavor of what we do by mentioning one enzyme there, TKLTL1, which is involved in energy metabolism, and this is work together with Wieland Huttner's group in Dresden, who are experts in brain development. So what we've sort of then done with this one change is to change it with CRISPR-Cas9 in human stem cells back to the ancestral Neanderthal-like state. So we change these stem cells, so in this one respect, they are now like the Neanderthals. And these stem cells you can then differentiate into small brain organoids, so little cell balls that you have in tissue culture that mimic early stages in brain development. So we can then compare an ancestralized such brain organoids with modern human-like ones, and look in the epithelium where stem cells divide down here, migrate up in the epithelium and differentiate into neurons during brain development. And if we look at these stem cells here, we find that in the ancestralized ones, there are fewer of these stem cells that will eventually become neurons. And it seems to also happen because if we look in the neurons up there, we have fewer neurons in the Neanderthal state. So that looks super interesting. So sort of one big question based on this and on other work is sort of, was neurogenesis different in Neanderthals and Denisovans compared to modern humans? But we are really struggling with this, and there are a lot of caveats to sort of mention. One is that we're really studying early development here, and that may be compensated later on, during later embryonic and fetal development, so that it may not have consequences in the adult individual. Also, I think that many phenotypes will be polygenic, so many genetic variants will influence them. So a single variant in isolation like this may not tell the full story. And finally, there is this gene flow from modern humans, into modern humans from Neanderthals and old variants lingering around. So some present-day people will carry these ancestral variants. And that is the case in many of this list when we now get hundreds of thousands of complete genomes and beginning to get them not only from people in East Asia and Europe, but from other parts of the world. So in this case, it's particularly striking, and that's why I bring this example. If one studies genomes from just 50 people from Southern Africa and from 50 people in the Philippines, indigenous groups, you actually find that many of these variants do exist there in their ancestral form. Often, you can't read this, but often it's very rare, sort of 1% or less of the population, but not always so rare. And one example is this TKTL1, where the ancestral variant doesn't exist in the Philippines, but does exist in these African groups and in almost 30% of individuals. And this is even more extreme because it's on the X chromosome. So it means that men in this group, around 30% of men in this group, do carry only this ancestral variant, and as far as we know, they are perfectly fine. So does that then mean that this variant is uninteresting? And although we see this clear effect early in brain development, it may not mean that it is uninteresting. So if we take a step back again and sort of think about with our current thinking about this, I think that we need to take a sort of combinatorial view of what makes modern humans special. If you sort of think of how morphologists who study skeletons define Neanderthals, they say that they have a combination of features. They have this robust skeleton, they have an oblong cranium and so on, big brow ridges. But you can find individuals alive today that have just as big brow ridges as Neanderthals have. But you cannot find a person that have a combination of all these features. So it may be that we need to think about modern humans and also our brain development or physiology in the same way. Sort of saying that you're a modern human if you have most of these things in a modern derived state. But none of these things is in itself necessary to be a fully modern human. So that's sort of our current thinking of this. The challenge is, of course, still that probably there are some things that are more important than others in that list, some that may even be crucial, and those are the things that for the next few years we really would like to be able to look for and find. Finally, before ending then, I want to spend a few minutes on things that happen on the Neanderthal lineage. So we now have a number of high-quality Neanderthal genomes and a larger number of low-quality genomes, so we can begin to look for things also that seem to be present or high frequency because it's present in all the Neanderthals we know, but not in modern humans or very rare in modern humans. And some of these changes then are important and influence us today. I will just talk about two types of such changes, two examples. One example of these enzymes, for example, that are expressed in our liver and are important for how we digest or metabolize many drugs we take. So these enzymes, some people carry variants that clearly come from Neanderthals, are closely related to the Neanderthals we have sequenced there. And if you happen to be a carrier of this Neanderthal variant and you happen to take ibuprofen against pains and aches, then you're actually in a good situation because you have a much better effect. You have a much higher, longer half-life of ibuprofen in your blood, so you sort of enjoy a much better effect than if you're not a carrier of the Neanderthal variant. More seriously, if you take warfarin against blood clotting because you have an artificial heart valve, for example, you need to take a much lower dose to avoid potentially very serious problems with bleeding if you're a Neanderthal carrier. And the most striking example to me, as was hinted at in the introduction, came during the COVID pandemic, where, as you know, most people who get got infected by SARS-CoV-2 had very few symptoms or hardly any symptoms, but some people became very sick and some even died. And we know some of the risk factors, being male, being old, diabetes, what have you. But all these risk factors are not enough to explain why certain people became so very ill and many others did not. So we were peripherally involved in a very, very small part in an international consortium back starting in 2020, looking for genetic factors in people influencing how sick you get when you get infected by the virus. And in the summer of 2020, the first results came, and they were surprisingly clear. There was one big locus on chromosome 3 that influenced to a great extent how sick you become. So there were these sort of risk variants there, and when we then looked at that and put it together with the Neanderthal genomes, we were shocked to find that these risk variants clearly came from Neanderthals, but particularly close to our Neanderthal genome from Southern Europe, from Croatia there, whereas the protective variants were modern, if you like. So clearly this sort of risk factor has come over from Neanderthals and then exists in some people today. And for being a genetic risk factor, this was a very strong one. So if you were hospitalized in Scandinavia early in the pandemic with severe COVID and were not a carrier of the Neanderthal variant, you had a 7% risk of dying. If you were a carrier, you had about double that risk of dying. So about doubles your risk when you're infected. So of course, we and others are very interested in what's hiding in this region, because if we understood the physiology of this, we could perhaps treat severe COVID better. But unfortunately, it's a very complex region. There are at least 3 genes in there where there are good hypotheses suggesting what might influence this. There are 14 nucleotide differences here that you can investigate, and it's not a clear answer to that. But it's interesting to look at the distribution of this risk variant around the world. As typical for Neanderthal variants, it's absent in Africa or almost absent in Africa, but it's also absent in East Asia and quite high in South Asia. So you can sort of estimate from this frequency how many extra deaths we had in the pandemic due to this Neanderthal contribution, and it's clearly more than 1 million extra deaths we've had. But this distribution is also interesting. It's clearly something in East Asia that in the past have selected against this variant. We don't know what that is. You can speculate that it's corona epidemics in that part of the world, for example, but we don't know. It has clearly been positive to have this in India or in Pakistan or Sri Lanka, though. We don't know why that is. But we're beginning to learn that this region influences the response to many infections. So Hugo Seberg, who's studied a lot of this together with us, he noticed that if you look at this risk locus on chromosome 3 and go 1 million bases to the right of it in this thing, there is another gene there that some of you have heard about. It's very famous, CCR5, which is a co-receptor for HIV. So the virus that gives you AIDS binds to this protein on the cell surface. And people who lack this gene, who have a deletion on both their chromosomes, are actually resistant to being infected with HIV, couldn't care less if they're exposed. So when you look on people who are carriers of this COVID risk factor from Neanderthals, they express less of CCR5. So this locus here influences genes, so it's less expressed in your immune cells. And that then has a result that you look in 3 different cohorts, for risk of being infected with HIV is about 25% reduced if you carry the Neanderthal risk factor for severe COVID. So this risk locus is bad to have if you're exposed to SARS-CoV-2 because you have a bigger risk of becoming severely ill and die. But if you're exposed to HIV, it's actually a good thing to have because you have a less risk to being infected. So as many such genetic variants, I think they're a double-edged sword. They sort of depend on the environment. In this case, what viruses you're exposed to, if they're bad or not. And of course, it's not just HIV and SARS-CoV-2. There's something else that influenced this in East Asia in the past and in South Asia. So it's sort of in reality a multi-edged sword, actually. And just before ending, I want to say that Neanderthals should not just have a bad reputation when it comes to the recent pandemic. When one now have more individuals or more resolution, you find other loci that also influence the result when we get infected by SARS-CoV-2. For example, there on chromosome 12. And in that case, one version there comes from Neanderthals, but in this case, it's actually protective and makes you less likely to become severely ill. We know in that case the biology behind that. There are sort of enzymes that come from Neanderthals there that sort of make this little signal molecules that activates an RNase that degrade the double-stranded RNA from the virus. So it's also protective against other double-stranded RNA viruses. But unfortunately, the effect size is much smaller. So the increased risk about doubles your risk to be severely ill, but this other one sort of decrease it if you have it from Neanderthals by around 25%. So this is just to give you a feeling for that there is still an influence from this gene flow from Neanderthals that can be very surprising and pop up when something new happens, such as the SARS-CoV-2 pandemic. And I hope that sort of convinced you then that it can be interesting to have the genomes of our closest evolutionary relatives, because we can look at things that are specific to modern humans and try to understand that, and we're just at the beginning of that. We can study things that are unique to Neanderthals, particularly those things that come over to us and have influence today. And the way we do that is then to use biobanks, where we can look at association with Neanderthal-specific things. But we also do experiments then to understand this more in detail, for example, in stem cells that we modify, or even sometimes, I didn't talk about that, in mice that you can Neanderthalize and modernize, if you like, to study the effect of these things. And most of this work then goes on in Leipzig, as we heard, at the Max Planck Institute. That's a recent group that works on these things, and a small group also in Okinawa in Japan that study particularly the mouse models for these things. And with that, I then thank you for your attention.
Thank you, Professor Pääbo. Thank you, Professor Pääbo, and congratulations again for your award. To conclude, Carles Marigó performs a piece created around Til Rosorna, To the Roses, by Wilhelm Petersen Berger, inspired by the island of Frösö in northern Sweden. Its inclusion in this program is not by chance, in addition to being chosen by Dr. Pääbo. It coincides with the time of the year when the event takes place, in the midst of spring. Like the roses to which it pays homage, this piece blossoms briefly, intensely, differing as a space of calm and contemplation. Gràcies, Carles Marigó, per haver-nos regalat aquestes 3 peces que avui també han estat protagonistes aquí al Saló de Cent de l'Ajuntament de Barcelona. Now, we have reached the end of this ceremony. Before we leave, we will ask those present on the stage to come to the center to official photo session. And we remind the other guests that the reception with refreshments will be held in the Galleria Gótica. Thank you all.